What's up guys, my name is Rohan Dursetti and I'm going to teach you about the life cycle of coronavirus. But to start, what exactly is coronavirus? Coronavirus is a new virus that first appeared in Wuhan, China in December of 2019, which is known to cause an infectious disease. It is commonly believed that the virus originated from bats and was transferred to humans. Moreover, infection is common through droplets because the virus can survive for 2 hours in the air. Anyone can get the virus, but those who are elderly or who have pre-existing medical conditions are more at risk regarding its effects. Looking at the virus itself, it infects humans when its surface spike protein, which is shown by the red corn-shaped spikes in the top left corner, binds to a human lung cell's ACE2 receptor, shown by the green semicircle overlapping it on the cell's membrane. This receptor is not intended for the virus's entry, but because it does control what is able to enter the cell, the spike proteins on the virus first make contact with this part of the human cell. The genome from the virus is then able to pass through the membrane, typically by membrane fusion. Once inside, it's worth noting that this viral genome has a 5' methylated cap as well as a polyadenylated tail, characteristics of an RNA strand. Because of this, the lung cell treats the genome as mRNA, putting the genome through the process of translation to ultimately produce a polyprotein, protease, and other accessory proteins. The accessory proteins help the virus bypass the cell's immune system and control gene expression. On the other hand, the polyprotein is cut by the protease into 16 parts, which come together to create an enzyme called replicase, which attaches to membranes inside the cell, typically the endoplasmic reticulum. The replicase makes an antisense copy of the genome to serve as a template for RNA duplication, and using this template, additional sense copies of the original viral genome are created. On another note, this demonstrates why replicase is known to be a drug target, which is a molecule that could be influenced by a certain drug to create a positive effect. If a drug can stop the replicase from duplicating the RNA, it can stop the virus from spreading. Drugs also may be necessary because although replicase has some screening ability, mutations to the genome occur specifically in the spike protein and accessory protein before it reaches a replicase. Because of this, the virus can infect our lungs while we cannot properly defend. Replicase also copies shorter parts of the genome as labeled by the mRNA pieces, which are used to build structural proteins. Lung cell ribosomes translate these shorter mRNAs into such membrane-bound proteins, which are then combined with a copy of the viral genome and are transported through the Golgi apparatus as shown by the small blue circular disc. This completed virus then exits the cell with the help of a vesicle fusing it into the membrane, where it is free to infect other lung cells, thus concluding the life cycle of a coronavirus. As a final message, please stay safe and healthy as we navigate through this coronavirus outbreak, and thank you for watching.